Hi, my name is Ayush Tambde and I'm a sixth-year student from Stratford College, Dublin, with my project titled Analysis of Single-Cell Proteomic Data to Understand Mechanisms of Oncogenic Transformations and How to Control them. But first, what does that mean? Activities in the cell are tightly controlled by communication systems known as signaling pathways. When these pathways mess up, it can lead to undesired side effects, such as cancer. In breast cancer, we generally have two broad categories: the relatively easy to treat luminal type and basal, which has a much higher mortality rate. If we can understand how these cancer cells change state, for example, see how luminal cells can turn into basal cells, it becomes easier to understand how we might be able to control these changes, or even reverse them. My project is about analyzing changes in various protein levels in both luminal and basal cells in response to perturbations which inhibit certain components of the cell signaling pathways, and then trying to figure out what changes in the cell signaling pathways can induce a change in the cell state. To analyze this dataset, I used the recently developed C-star techniques. First, I constructed a support vector machine. This is a machine learning algorithm which learns to partition data into two separate classes. In this case, it's luminal and basal cancer cells. Then, I used the results of this classification to calculate dynamic phenotype descriptor, or DPD values, which quantify how luminal or basal these cells are. I used an algorithm called Bayesian Modular Response Analysis, or BMRA, to reconstruct the core cell signaling network based on the DPD values and the original proteomic data. The signaling network helped me create a biological model using BioNetGen, which encodes more specialized biochemical knowledge, such as how certain proteins interact with each other, or how activation of one pathway can activate or inhibit the other. This model also describes Waddington's landscape, a particularly intuitive analogy where we imagine marbles rolling down a hill. Each marble represents a cell, and each valley represents the possible states that the cell can take. From the preliminary results of the analysis, we can see here that across all timestamps, the cell signaling component TGF-beta SMAD is quite strongly correlated with the basal phenotype, with a blue activation connection. We can also see that across all timestamps, there is a positive connection between TGF-beta SMAD and the MEK component. Looking at our proteomic data here, we can see that inhibiting MEK and mTOR also seems to inhibit TGF-beta SMAD, and that these treatments also lower the DPD value, meaning that the cell is pushed closer to the luminal phenotype between the 10 and 20 minutes time points. This suggests mTOR and MEK inhibition may be used potentially to transform cell states. Once the biological model is finished training, it will be easier to simulate and understand these transformations. Thank you so much for listening to me, and I hope you have a great day.